Okay, so like I was saying, we have to start a new topic today. This topic is nucleic acids, protein synthesis, and everything that surrounds what happens to your beloved cell's nucleus, right? Now, I told you already that that nucleus is responsible for replicating on itself to form another cell that's then going to divide in mitosis. That nucleus will obviously also divide in meiosis. That nucleus is going to give us the proteins because it is the nucleus that has genes. Just before I continue, can you guys hear me and see the screen?
Yes, teacher.
Yeah. Yes, okay, perfect. Yeah, I don't want to like sit here talking about the question like the other time and then get to know that you couldn't see it. So, okay. Now, so we know nucleic acids are responsible for everything, right? With that note, let's see what we are going to do today. Some of this today, some of this tomorrow, obviously. So he said, so the learning outcomes want us to describe the structure of nucleotides, the structure of ATP, the base about tell us about the bases. The structural formula are not expected, but genetic information is needed. Then we are supposed to know DNA, how it's organized, the bondings between it, and then semi-conservative replication. And finally, we need to end on the RNA molecule. And then there's different kinds of RNA molecules. So it's going to be mixed throughout, and let's see how it goes. Again, like always, wherever you don't get something, just ask me at that point. Let's start with a nucleotide. Nucleotides are simply a sugar, a base, and a phosphate. We did this in topic 1 as well. In topic 1, I showed you pictures in those monomers. I showed you that there's something called a deoxyribonucleic acid, or the DNA as you know it, or ribonucleic acid, or the RNA as you know it. It's a polymer just like any other polymer, like polysaccharides and proteins, but its base unit is different. Its base unit, or its monomer, is a pentose sugar. This is also where I told you that sometimes, well, not sometimes, most of the times, we have hexose sugars, the hexose sugars with the 6 carbons that are in sucrose, maltose, etc., etc. But here we have a pentose sugar. You can see the 5 corners, and it's pentose not because only of the 5 corners, but also it has 5 carbons in it. If you remember, one other sugar was pentose shaped, but it still had 6 sucrose is made up of, and sucrose is made up of it. Okay? So it still had an extra carbon hanging over here, right? But that's not the case here. It's a pure pentose sugar with 5 carbons. This sugar has one base and one phosphate attached to it, and that is the entirety of the nucleotide. Now, these are the bases that are the adenosine, etc., etc. We're going to see them later, but for now, a pentose sugar, a phosphate, and a nitrogen-containing base. So the next time somebody asks you what monomers or what polymers contain nitrogen, your answer is not only going to be proteins, your answer is also going to be nucleotides or DNA, RNA, because this base contains nitrogen as well. So 3 things. Remember these 3 things. So moving on, okay. So now we have the DNA and the RNA. Now we'll go step by step. First, let's talk about the bases. For the bases, stay with my pointer here. For the bases, what we actually have. So before the bases, let's start with our pentose sugar. For our pentose sugar, you can appreciate these 5 carbons, 1, 2, 3, 4, 5, and obviously the O of the carb sugar is always within the ring. So the O is over here. Both RNA and DNA are made up of nucleotides. DNA is deoxyribonucleic acid, and ribonucleic acid is RNA. One of them is called a nucleotide. This one is a nucleotide. When many nucleotides join together, they form nucleic acids. Okay, that is much for now. Now the pentose sugar, both have a pentose sugar. Deoxy has a deoxygenated pentose sugar. Ribonucleic has oxygen. It is not deoxygenated. As you can see here, look at the second carbon. Look at this over here. Everything else is similar except that this is deoxygenated. The oxygen here is hydroxyl oxygen is removed. We only have a hydrogen. However, here we do have a full hydroxyl group. Other than that, the bases are also, phosphate is obviously similar. The other thing that's different is the bases. So generally speaking, when we say bases, we have three bases that are common between them and two bases that alternate between DNA and RNA. And then we have the three bases that we have is cytosine, guanine, and adenine. Cytosine, guanine, and adenine, names that you need to remember for lifetime. And then there are two other bases called thiamine and uracil. Now, as you can see, thiamine and uracil have they're exactly the same thing, except thiamine has this CH3 here that uracil does not. But they're the same thing, and thiamine is present in DNA, whereas uracil is present in RNA. So what are the four? So each DNA, whether it's DNA or RNA, has four bases only. DNA has thiamine, cytosine, guanine, and adenine. RNA has uracil, cytosine, guanine, and adenine. These are common bases, whereas these are that make the DNA or RNA different. Okay. The second point you need to know over here is that two of these bases are larger than the other two. For instance, these are double ring bases, which means if I'm representing each base by this strand over here, this one yellow strand is representing adenine, and this pink one thiamine. Okay, no, so the pink one's representing uracil because it's in the RNA, right? The color coding isn't quite right here. So if this is one base, then the smaller bases are the single ring ones, uracil or cytosine, and the larger bases are guanine and adenine, larger in length because of the double ring. So guanine and adenine are the larger bases, and these larger bases. So I know you guys have read this topic. There's difference between pyrimidines and purines, right? Two of these bases, the larger ones, what are they called? Anyone?
Pyrimidines.
Purines. The larger bases are called purines. The smaller ones are ironically called the longer word, which is pyridines. Okay, pyrimidines. So that's the other difference. So on this, it's a very high yield slide. You need to appreciate DNA and RNA. I've made you appreciate the difference in the pentose sugar. I've made you appreciate the difference between uracil and thiamine. Second thing that you appreciate is the bigger double ring ones are called as purines, smaller ring ones are pyrimidines. And because whenever we want base pairing to occur, and what is base pairing? Base pairing means, like I told you, hydrogen, whenever attached to something like nitrogen or oxygen, develops a polarity. Base pairing is the attraction of this base with another base. See, this base here is attracted to this one. Hydrogen bonds form for DNA to achieve its double strands. So what you need to understand here is that this structure of the DNA, what ends up happening is the sugar and the phosphate remain as they are. And what happens is for this nucleotide to become a dinucleotide or a trinucleotide, which means 1, 2, or 3 nucleotides, the sugar and the phosphate will form their own bonds here. And then that's going to make it a nucleotide, an RNA. Okay. This RNA is single stranded. RNAs are always single stranded. DNA is double stranded. And it doesn't matter, even DNAs can be single stranded, as we can see in the virus, in some viruses. But what is important to remember here, and that you'll appreciate later as well, is that the original structure to form a nucleotide is not base pairing. It is this over here, this ribbon-like structure, which is formed due to our phosphate and this ribose. It is not formed due to base pairing. Base pairing is only for two similar strands to join together. RNA does not necessarily in a eukaryote is usually is single stranded and DNA is double stranded, but in viruses, RNA can Also, there's one specific virus with a double-stranded RNA, which means why do we need base pairing? Why do we need these bases to form hydrogen bonds due to this polarity to form double strands, right? For one nucleic acid to form, all we need is this deoxyribose or ribose and the phosphate group. And the colored stand here are the fancy beads, and fancy beads attract other fancy beads. And so in that case, the mnemonic that most people use is at GC. Let me write it down here for you just so you guys know what I'm talking about. So there's a mnemonic that we use to understand base pairing. And when I say base pairing, what I mean is that if this was a, let's say, DNA, I'm supposing here, and if I want a base pair hit, and if I say this is adenine, then there's going to be another base over here. This base is going to form a hydrogen bond over here for this to remain attracted together, right? This is exactly what's happening over here. These hydrogen bonds form. So the mnemonic we use is called at GC. And these are the lines I'm drawing between them. Now, what is this supposed to show you? It shows you, like I said, because I want the diameter of the strand to be equal throughout, it means if one large base forms bond with a smaller base, it makes sense, right? It cannot form bond with another large another large base, otherwise large kinks will exist between them. So what pairing takes place? Pairing always takes place of an adenine with a thiamine in DNA. And if it's an RNA, then of course, thiamine version of an RNA is always uracil, right? So that is understood. You don't have to learn it. Thiamine, like I told you, uracil, thiamine are the same thing. In the RNA, it's called a uracil. And what do these lines show? These two lines show that double hydrogen bondings take place. This hydrogen and this hydrogen bonds with the thiamine. So adenine and thiamine at GC. GC has triple hydrogen bonding. You can see the three lines, and the G stands for the guanine and the C for the cytosine. Again, a double ring with a single ring. And then again, because of the so many larger number of nitrogens, look at this here, there's almost 6 nitrogens over here and 3 over here. So this overall gives us 3 bonds, whereas this one, no matter how many nitrogens over here, this molecule only has 2 nitrogens. Thiamine and uracil have only 2 nitrogens. So 2 nitrogen bondings are possible. So at GC, mnemonic to remember which purine binds which pyrimidine and which pyrimidine binds which purine. Okay. Again, please make sure that you understand and appreciate this slide well enough. Any questions on this slide? No, teacher. Okay. So again, the image over here, just to show you the DNA and RNA, the base pairing, the types of bases. Yeah, that's basically it. Okay. So let's move on to the next slide and see what we have next. So again, he's showing you the differences. These 3 bases are common. This one is different. This one is different. This one has a ribonucleic, a pentose sugar, a deoxyribonucleic, always single-stranded, always double-stranded. And then the bonding is obviously at GC. GC has 3 bonds, at has 2 bonds. Okay. Again, very clear. Just to reiterate the point I've put on the slide. So now we're talking about ATP. ATP is a modified nucleotide. It is used to deliver energy. The energy in the ATP, so if you minus out these 2 phosphates, you see that it's just a ribose and a base with 3 phosphates, right? So what's basically happening is ATP is a very specific molecule that provides energy. It has 3 phosphate groups, out of which it loses these 2 phosphate groups to provide energy and keeps 1 phosphate group because obviously it has to retain its nucleotide properties. Now, the specific thing about ATP is it's called adenosine triphosphate, and the name gives it away. This ribose sugar combined with adenine is called adenosine. Look at this here. And then triphosphate. Similarly, if this ribose sugar is combined with a guanine, it's called guanosine. I'm not talking about the phosphate yet. If it said GTP, it would be 3 phosphates, guanine with a ribose, making it guanosine triphosphate, right? Again, thiamine with a ribose sugar. If I put thiamine over here, it would form thymidine. Okay, uracil over there, uridine, cytidine, again, adenine, adenosine, guanine, guanosine, and then the triphosphate or diphosphate, or even sometimes it says AMP. The basic form is adenosine, this molecule, both of them combined, adenosine plus monophosphate, adenosine plus diphosphate, adenosine plus triphosphate. So that is your ATP. All there is to know about ATP. The only thing that you need to know other than that is that sometimes he may say, okay, ADP plus PI gives you energy or it gives you ATP. What he means is that you have an adenosine, this molecule, both of these, and then you have a diphosphate, you have two of these. And this phosphate, if for instance it wasn't here, and then adding to this adenosine diphosphate is an inorganic phosphate molecule, PI, inorganic phosphate molecule, that is going to attach over here to form this ATP. Again, adenine, ribose, triphosphate, this adenosine. All of these combined, adenosine triphosphate. Clear?
Yes. Yeah.
Okay, great. Wherever you guys don't understand something, please do point it out, okay? Because this is a relatively tougher topic as compared to the previous ones. It's completely biochemistry. Okay, I hope we are clear. Right? Are we clear on your end as well? Okay, perfect. Okay, let's move on and see what do we have next. Okay, so next, it's the book version of telling you that there are complementary base pairing at, like I told you, GC with three, look at the appreciate, there are 3 lines between here and 2 lines between here. Again, he's also telling you that one complete turn gives us 10 base pairs. He's showing you the backbones. What's the backbone is basically what's binding together to form the original structure of the nucleic acid. And he's telling you that because the base pairing is involved in complementary bases to join 2 of these nucleic acid strands. So the backbone or the original structure is the sugar and the phosphate, the ribose and the phosphate, right? And then the center, we have the bases and we have 2 anti-parallel chains. Now, what is that supposed to mean? So let's try to understand what that's supposed to mean. Okay, so over here, when we're talking about this sugar, now guys need to understand this very clearly. So I told you there's an O over here, there's a carbon here, and whenever we are in a sugar molecule, we always count the carbons. Where do we count the carbons? We always count them with clockwise, right? Okay, so we have a carbon 1 here, a carbon 2, a carbon 3, a carbon 4. The 5th carbon goes over here, which is attached to the phosphate group, right? 5 carbons complete. It's a pentose sugar, 5 carbons, 5 corners. It's C is equals to 5. Okay. Okay. So now I told you if this is one of the molecules, and I need to, and we've been doing this commonly, if I want you guys to make a polymer out of it. So you guys, I don't know if you've attended my topic too, but we made polymers, right? And for polymers, what we did were we made bonds. For instance, we made a bond of a peptide and we said, okay, this is the peptide bond. And then we joined them together and we said, okay, fine, this is the peptide bond. Now, if I ask you to make a nucleotide bond, what are you going to do? Now you can't assume it because it's tougher than the normal bond. So I'm just going to tell you that over here. So this is my ribose sugar. This is my base. I know that these bases are involved in their own pairing. So this molecule, this part of the equation is involved elsewhere in hydrogen bonding. Now I have this part that we keep calling the phosphate or the phosphosugar backbone, the phosphate sugar backbone. Why do I call it the phosphate sugar backbone? Because there's a special bonding called a 3,5 phosphodiester linkage that takes place between these molecules. Now, what is ester linkage? Ester linkage is basically between these carbons and hydroxyls. Okay, and then we have phosphate involved in the linkage as well. So if I tell you simply, it's going to be between 3s of one molecule and 5 of the other molecule, right? So what ends up happening is, for instance, I told you that the carbon of the 5 is over here, right? Can you appreciate over here? Attached to the phosphate. What ends up happening is this carbon number 5 with the phosphate in the center comes below this nucleotide and forms a bond, keeping the phosphate in the center. It forms a bond with the carbon over here. What was the number of the carbon over here? 3. So when this bond, this CPC bond forms with 1,5 and 1,3, it's called a 3,5 phospho. And because of the hydroxyls of the carbon, diester bond. So is this bonding clear?
Yes, teacher. Yeah.
So I try to show you again as well. Wait, let me see if I can show you over here. Again, can you appreciate? Look at this one here, carbon 1, 2, 3, 4, and carbon 5. And this carbon 5 is attached to a phosphate, like I said, and obviously this carbon 1 is attached to the base. And so when this phosphate goes in, and this phosphate has a very special structure. If you notice the phosphates here, appreciate that they have in fact, what they actually have are these hydroxyl groups. Can you appreciate it's a whole hydroxyl group. I mean, it's an OOH, just like a carboxyl group. So it's one of those, a carboxylic acid, like the carboxylic acid group, right? And so what is the ester linkage? Ester linkage, basically, if you remember from organic chemistry, it was this linkage, this carboxylic acid plus an alcohol, right? So if this is the carboxylic acid part, and every C we saw had an OH just like this one. So if I say that with my next molecule, it's 3 number, this hydroxyl from this 3 number will bind to this phosphate with its carboxylic acid, which is bound to this 5 number. It's going to be a phosphodiester linkage, right? Why is it diester again? Because of this carboxylic acid and this alcohol binding together, okay, with the phosphate in the center, because it's more convenient that way, obviously, for it to, okay?
So wait, there are two ester bonding? That's
yeah, there are two ester bonding because one on the top, one on the bottom. Because this 3 has to bind with something at the bottom as well, right?
Yeah.
So there are diester linkages taking place to form these, okay? So there's 3, 5, and even if you don't understand it, because it's biochemistry and this is we just have to do the biology part, like this said, that structural significance is not needed. I told you for your convenience, even if you can't understand it, just remember 3, 5 phosphodiester linkage, and it's going to become clearer as we move forward, okay? Just give it a little bit time. Okay, now look at this. I'm talking about a diester. Look at this. Number 5 sugar with the phosphate in the center attached to the number 3 sugar to give this dinucleotide. And if I want an entire chain, then you can appreciate there's this 3 sugar over here that is bound to something below it, or even if it's not bound, what if it's just empty, right? And then there's phosphate group, and then this phosphate to this 3, the 5 sugar phosphate, this 3 again, 5 sugar phosphate 3, 5 sugar phosphate 3, 5 sugar phosphate 3, until we reach this 5 sugar, which I say is not bound to, for instance, anything. So what I want to teach you over here is that at the end of the chain, we have either a three number of carbon, 3 numbered carbon remaining, or a 5 numbered carbon remaining. Now, what does that mean? What that means is that one nucleic acid strand runs from 3 prime denotes the number of the carbon, to a 5 prime, or a 5 prime to a 3 prime, whatever direction you want to say. For instance, if I say the base I put here is adenine, okay, and the base I put here is cytosine. Now, if somebody, Abdullah, standing here and reading from this side, now the Abdullah is standing on the red dot, and I ask him, what do you see? He's going to tell me, okay, I see if 5 prime, my nucleic acid is starting from adenine or an adenosine, running from 5 prime end to 3 prime end. What does that mean? It means it's starting from the 5 prime end and ending at the 3 prime end. And now I'm standing here and I say, no, mine is starting with a cytosine running from a 3 prime end to a 5 prime end. So in fact, your movement or your direction or where you're standing determines which prime end you are at. For instance, over here, let's look at this over here. If I'm standing over here, what am I going to say? That my nucleic acid is running from what prime to what prime? If I'm standing over here.
From 5 to 5.
Okay, I'm standing over here. What prime do I see? What prime do I encounter that's first? A 3 prime, right? Not a 5 prime. 5 prime is over here. I encounter a 3 prime. Then I see this 5 prime that is bonded. And then I see a free 5 prime at the end. So if I'm standing over here, I'm going to say, okay, the first thing I saw was a 3 prime. So the opposite end, as you can see, is always going to be either 5 or 3. So it's never going to be the same. If I start from a 3 prime end, the other end is always going to be 5 prime. That is in the notebook. That's a fact. Okay. So if I'm standing here and the first thing I see is a 3 prime, I don't even need to look at the end of the chain. I know that that chain is going to end on a 5 prime now. And if I'm standing here and I see a 5 prime, then I know that chain is going to end on a 3 prime. Okay. So that's and why was I telling you this whole story? Because when you look at this DNA, which has formed these pairing between its bases, it says 2 anti-parallel chains. And what does it mean by these anti-parallel chains? What it means is that if we're both standing here and we're looking at this chain, if it goes from 3 prime to 5 prime, then the chain that will form base pair with this chain will always be its opposites. It will be from 5 prime to 3 prime. Do you get what I mean? What I mean is, if we are both standing here, you're standing here, I'm standing here, and I'm looking at this chain, if I see, I do not need to see the other chain to see its direction. If I see this chain is going from 3 prime to 5 prime, the other chain is going to be its parallel. It's going to go from 5 prime to 3 prime over here. It's going to come in the opposite direction. If I say that if I call the correct direction of a chain to be 3 prime to 5 prime, then, and I also say that now I just told you that if it starts from 3 prime here, then its base paired chain will start from 5 prime here. It cannot start from 3 prime. That's a law. Then this will end in 3 prime, obviously, which means if the original way of the bus to run is from station number 3 to 5, then this bus is going to run from 3 to 5 as well, which means these are anti-parallel to each other. What are parallel lines that run in the same direction? What are anti-parallel lines that run equally but in opposite directions? So is this clear? What are anti-parallel chains?
Yeah.
Yes.
Okay, so if I say I was standing here and you were standing over here, and I ask you in what direction are the trains running, you say, okay, this one's running 5 prime to 3 prime, 5 prime to 3 prime, right? And I ask you what direction the other one's gonna run. So it's gonna be anti-parallel. So now you know it's anti-parallel, it's gonna run this way, which means its arrowhead is going to be at the 3 prime, which is here, which makes sense because 5 prime always coincides with the 3 prime. 5 prime, it's like opposites attract each other. 5 prime will attract 3 prime and 3 prime will attract 5 prime, and there is no way around this, which is why they're called anti-parallel chains. They are parallel to each other but anti in direction. Okay, so I hope this is clear. Is it clear for you too, right? Everything going so well so far? Okay, so you guys have read this before. Understand it from the perspective of clearing your confusions and not being introduced to the topic. Okay, so is it clearing your confusions yet?
Yes, teacher.
Yes.
Raid hasn't read it, so it's a first time for Raid. As a first time, are you understanding it? Do we need to go into more details, or is it becoming clear this way? I don't want to go into more details of biochemistry because the syllabus clearly says that you don't need it. So I just want to make sure that you understand what's happening. Okay, perfect. So let's do this. Now, very interesting table. Now what you need to understand is the purpose of this table. So when I'm saying two anti-parallel strands, does this mean, what does this mean? It means so when I say anti-parallel strand, what it actually means is that these base pairs, it doesn't mean that these base pairs will switch. It means these base pairs will follow their own direction at GC, right? So at GC, which means if adenosine here, then thiamine over here in the case of DNA, or uracil if it was an RNA. Again, I said cytosine here, so guanosine here. Now, what does that mean? It means that if I know that the number of adenine and thymine is going to be similar, right? Because if in this DNA, I say that in this strand number A and strand number B, if I say strand number A had 60 adenine, had 30 adenines. So if strand number A had 30 adenines, and adenine can only form a bond with thymine, which means thymine are also going to be 30, right? They cannot be more.
Yes.
Okay. And then if I say, okay, both of these together, adenine in this DNA molecule, the complete double-stranded DNA molecule, we have 13 adenine, which means we're going to have 30 thymine, which means total 60 of these. Then I have 40 more left from 100%. I'm talking about percentages right now. Okay. Because these are not quantifiable units. Whenever we do PCRs or other analysis to check for base pairing, they're always by percentages amount, never by concrete amounts, never by arbitrary units. Okay, so 60% is adenine and thymine combined because both of them will be equal to each other to bond, right? And then we have 40% remaining. Now this 40% is going to be both G and C, right?
Yes.
So how many of this will be G and how many of this will be C?
20-20 each.
20-20 each. Very easy because these 20 have to bond to these 20, right? And this table tells you exactly that. For instance, we checked a human cell and we said, okay, adenine was 40%. So thymine, so how much, if this was 40%, then how much should thymine be? Thymine should also be 40%, right? And then we checked again and we said, okay, if guanine was 20%, then cytosine should also be 20%, right? And then in that case, we then look at the differences that are happening to determine if there's a problem going on or what's going on. Sorry, this isn't 40%, this is 30%, okay? So if you can see in percentages, both of these are equal. It's 29.4, it's 30.9. Very little difference. Both are 30%. 19.9, 19.8. Very little difference. How similar are these? Again, let's look at E. coli, the bacteria. We saw that even in bacteria, look at how closely related these are and how closely related these two are, which shows that AT always pairs with this and GC always pairs together. So the whole purpose of this table is to show you that both of these will always pair together, never alone or never with another one. Okay, this is standardized. Some things in DNA are always standardized. The 3' to 5', the opposite base pairing, how adenine will always be with thymine, how it will only have 2 hydrogen bonds always, G with C, 3 hydrogen bonds always, the anti-parallel strands. So yeah, that's basically about it. This one is showing you the rings again. Look at this, the double hydrogen bonding, the triple hydrogen bonding, and then these are the base pairs. Now appreciate the ribose and the phosphodiester bondages. So let's see what these are. Okay, look at this one. So look at this one. This is the 5' end, the phosphate end, and the 3' end, right? Because it's always 3 and 5 bonding and nothing else. So if I was to label where it's coming from and I'm standing over here, let's label one strand and see the other. Which strand do I see free over here? Okay, so this strand, let's go about it. We have to see the bonding, right? If this is the O, and we always have to look at the generic structure. So if this is the base pair, right, then this definitely means that this one is binding. This one's the phosphate right here, which means the 5' end should be somewhere over here, right? Because if you remember the general structure, what was the general structure? We had the phosphate over here and the base pair over here, right? So if I see the base pair, then I see, okay, 5' structure somewhere over here. So I definitely encountered the 5' structure first, and then this will be binding with the 3' structure. Okay. But I honestly, they're not going to ask you this because this is, again, very complicated biochemistry. So even if you don't understand it, it doesn't matter. This is for your own clearance. But one thing you need to understand is they're always going to tell you. So if they tell you this is running from 3', it's 3' over here, and they ask you to fill in the blanks. So you're going to write 5' over here, and the other strand is going to be the exact opposite. That's what they're basically going to test you about. They're never going to test you about naming them yourself. And this is a molecular image they've drawn just to show you how, look at this, DNA is coiling upon itself. Okay. So, and this is so this one is just to make you appreciate how AT have double, have 2 hydrogen bonds, and GC have 3. Okay. Again, look at this, look at the, okay, so look at the 3 to 5'